Hello, everybody. My name is Daniel Tamarit, and I would like to start by saying that I'm extremely happy to be able to contribute to this event. So thanks a lot to Valerie, Deanda, and to Joe Shuttleworth for the invitation. Um, so yes, I'm a postdoc at the Laboratory of Thijs Ettema in Wageningen in the Netherlands. And I'm going to talk about the incredible growth that has occurred in the microbial tree of life and how this has impacted our understanding of the evolution of cellular complexity. So in order to visually introduce the tree of life, I'm going to start by quoting Carl Sagan. In his show Cosmos, he said that there's an unbroken thread that stretches from the first cells to us. And I think this is a very powerful metaphor to see how we directly descend from some of the primordial organisms in our planet. And it will also be very helpful to visualize how all species on Earth are actually related. So in order to see this, here we have a stick person. So this person represents the whole of humanity and the entire human species. And this person is holding the end of a thread. So this thread will go back in time and we're going to follow it. So very soon, of course, we would find the last common ancestor of all humans. But then if we keep on going generations after generations, some millions of years ago, we would find the last common ancestor of all primates. But we can keep on going, and then we would find at some point the last common ancestor of all mammals, and then of all animals. So we are already some 500 or so million years back in time. But we can keep on going, and around 2 billion years in the past, we would find the last common ancestor of all eukaryotes. So eukaryotes are organisms that have cells with a nucleus and mitochondria, and they are going to come up again quite centrally in this talk. But yes, we can still keep on going, and some 4 billion years in the past, we would find the last common ancestor of all living beings and the first cells related to our lineage. So as you can see, this was a very, very long journey of around 4 billion years. And this really comes to show how a currently living species directly descends from one of the original organisms in our planet. But of course, this is an exercise that we could have done with any other currently living species. And if we would do that, we would see that each one of those species would hold the end of a thread, but the other end would always be the same. So these threads would interconnect and interweave, and together they would form what we call the tree of life. And the tree of life, of course, contains all species that live and that have lived. And we can easily find some that we are very familiar with. Like, for example, here in green, you can see plants and algae. In yellow, you can see fungi. And then in orange, you see all animals. And these three groups contain basically everything that we can see with the naked eye, all organisms that we can see with the naked eye. Everything else is majorly microbial. So you can really see that the tree of life is mostly a microbial tree. And then if we look at it with a global perspective, something that we currently do in order to classify all current life is to divide into three different categories. So here in pink, you can see the eukaryotes, cells with nucleus and mitochondria. And then the rest of the organisms would be divided into two different groups. So we have on one hand the bacteria here in blue, and then the archaea here in yellow. And the tree of life, of course, is extremely diverse. And you can see that the bacterial and archaeal side of the tree are hugely diverse as well. But this is the diversity existing in this tree is something that we have only started to really glimpse in the last decade or perhaps two. And before this, we had a very, very reduced knowledge of the lineages that exist in our planet. And it's advances in massive sequencing that have really turned this upside down. So one already classic example of this is this figure published in 2016 from Laura Haag et al. In a paper in Nature Microbiology, where the authors showed the diversity known at the time. And here you can see the lower branches here in green, eukaryotes, and then the rest of the lower branches would be archaea. Then everything else in the upper part of the tree would be bacteria. So here you can see this purple group that the authors named the candidate phyla radiation, and this included a lot of newly discovered lineages that we simply had no idea existed. And you can see by comparison with the rest of bacteria, this represents a huge chunk of the tree of life. And of course, additional lineages within bacteria have been discovered in the last decade or so. And this is also true for the tree of archaea and within eukaryotes as well. So in order to describe this in archaea a little bit more, you can see this figure from a review from 2017, where the authors described how the archaeal side of the tree of life developed in the last decades. So yeah, in the 1990s and early 2000s, we knew of only two archaeal groups, the Euryarchaea and the Crenarchaea. But then the following years were very informative in showing that really we didn't really know much about archaeal diversity yet. And in this coincided with the years that I started to study biology. So for the first time, I understood what archaea were. And then every year of my studies, one archaeal lineage, one phylum or equivalent group was described for the first time. So that was really mind-boggling to me, because it seemed that very late in biology, we were starting to understand diversity on Earth. But indeed, it was only the beginning. And here you can see in the bottom half how the tree of archaea looked like by 2017. And there's many additional lineages that we simply didn't know before this time. So for example, here you have the Deepan archaea that before we only knew one lineage, the Nanoarchaea, and yeah, everything else was unknown, and it has happened to be a very interesting and diverse group of archaea. And here in pink in the lower right side, you have the Asgarakia. So Asgarakia are a fascinating group. I'm going to focus more on them. And yeah, you will see how their discovery has also not only revealed that we knew very little about the diversity of archaea, but also helped shape the tree of life a little bit better. So in order to talk about Asgarakia, I'm going to present the efforts that led to their discovery. And it all started with a group of researchers choosing to explore the microbial communities existing in the bottom of the ocean. And they sampled the ocean floor in the North Sea, close to a structure called Loki's Castle. And of course, they found a lot of interesting microbes, but one that really stood out was what was called Loki archaea. And Loki archaea were fascinating in several regards, but of course their most salient feature was that they seemed to be the closest relative to eukaryotes that was known by the time. So the origin of eukaryotes is a very complex field that has suffered a lot of controversies and has focused a lot of attention of researchers. And something that was established by the time was the fact that the ancestor of eukaryotes originated from two different sources. So there was a symbiotic event of two different organisms, and their fusion and merger basically originated eukaryotes. So these ancestors were an archaean on one side, and then on the other, some bacterium somehow related to an alpha proteobacterium that originated mitochondria. But very little was known, particularly about the archaeal ancestor of eukaryotes. And the discovery of the Loki archaea, which was published in 2015 for the first time, was the first glimpse into an identity, single lineage of archaea that was more closely related to this archaeal ancestor of eukaryotes, and can therefore tell us something about their biology and their origin. So again, this was 2015. This is a very recent discovery. And the following years were tremendously informative in showing that Asgarakia collectively, so this is Loki archaea and close relatives, were a pretty diverse group. And indeed, there were two publications from 2016 and 2017 that found at least three major groups related to Lokiarchaea, and that were called Thorarchaea, Odinarchaea, and Heimdallarchaea, of course, keeping with a very nice theme, and collectively called the Asgarakia. And yeah, so phylogenetic studies showed that these Asgarakia collectively seem to be the closest relative to eukaryotes, although the exact positioning of eukaryotes with respect to these was still very undefined. And of course, their genomes contained a lot of fascinating information. And indeed, they seem to contain many genes that were thought to be typically eukaryotic. And these were related to several different functions. There were some genes related to N-glycosylation in eukaryotes that were present in Asgarakia, as well as escort machinery, for example, that has to do with membrane remodeling, ubiquitin system, cytoskeleton, or trafficking machinery. So these were somehow building blocks for typically eukaryotic functions that were already present in the Asgarakia, although of course the discovery of these was only done by informatically, so it's not very clear what kind of functions these genes perform in Asgarakia. Although in the last couple of years or so, there's been some very interesting experimental studies showing that at least for some of them, such as some cytoskeletal proteins, they do indeed seem to have similar characteristics as the eukaryotic homologs. But indeed, very little is known about what these genes do in Asgarakia and what role they may have had into their origins. So yeah, not much is yet known about Asgarakia, and one effort that we are currently undergoing to characterize them a bit better is a research project that we are carrying at Isetema's lab that I'm doing in collaboration. Well, it was originally led actually by Laura M. and Eva Cáceres, and we're working on this together currently. Where we expand by quite a lot the set of genomes that was available for Asgarakia from the original 11 from the 2017 paper, although there's been additional publications with more genomes in the last years. And then in total, we have now about 80 genomes that we are describing and characterizing with an evolutionary biology framework. So these 80 genomes, they are very diverse, and there's a lot of information that we are trying to extract from them. And several of them come from our lab, but there's been amazing work coming from some collaborators that were able not only to reconstruct a lot of genomes, but also find very interestingly diverse ones. So together, this forms a very nice data set to understand a bit better the genomic diversity existing in Asgarakia. And indeed, we find a lot of genomes related to the groups that I presented previously. So here in the darker shade, you have the proportion of genomes that was known by 2017. And then this study expands by quite a lot, each one of those. But additionally, we have found several major groups within the Asgarakia that we didn't really know about before. And we have called them Freya, Iden, Hel, and Balderarchaea. Indeed, the Helarchaea were published last year in a Nature Communications paper. But indeed, together, these set of genomes has a lot of very interesting information that, it's taken a bit of time to analyze. And of course, part of this effort has to do with trying to reconstruct a trustworthy tree of archaea and eukaryotes and try to understand what the archaeal ancestor of eukaryotes look like. And something that we consistently see in our phylogenetic analysis is that most Asgarakia seem to form a single monophyletic group, while there's a couple of groups, the Iden and the Heimdallarchaea, that are closer related to eukaryotes. And in this group, we don't really know exactly what the actual topology is, whether eukaryotes are more related to one of these two groups or perhaps to both equally. But this topology of Asgarakia into two major groups seems to be quite consistent in our studies using different gene sets and different phylogenetic methods. Yes, so there's a lot that has to be still uncovered about the Asgarakia, several additional very interesting genes that we are analyzing currently, and yeah, I urge you to follow this this topic, and hopefully we will be able to wrap it up soon for publication. We'll see. I also wanted to mention that this growth of the tree of life is coming from many different places, and indeed at the bottom of the ocean, Loki, Archaea, and the rest of the Asgards are far from being the only novel lineage. And other studies performed at a similar time in our lab from other samples from marine sediments, they also uncovered very unusual, very interesting lineages that raised a few eyebrows. And these had to do, well, these were related to the Chlamydia. So Chlamydia are well-known bacteria that infect mostly animals. Those are the ones that we know most about. There's also a lot of Chlamydia that infect protists. And indeed, here you have a tree of Chlamydia that we reconstructed recently. And in color, you see the Chlamydia that were known before this study, and in orange, Chlamydia that were newly described. So this was work that was mostly performed by Jenna Duramshi. She's a great PhD student in Theis's lab. And this was published earlier this year. So yes, indeed, Loki's castle also contained a huge diversity of Chlamydia that didn't really seem to be related to the ones, well, like they didn't seem to be the ones that we know about, but they expand by a lot the diversity of Chlamydia as we understand it now. And indeed, for many of these, there's still a lot that we don't know. And there's indications that some may not even be host associated, which would really be a fascinating discovery and help us understand the evolution of pathogenicity and host association. And indeed, one of these groups, we have called it the Anoxychlamydiales, and they are extremely interesting. And in a follow-up study that was led by Jenna and also Courtney Stairs in our lab, so they have analyzed a group of genes from this Anoxychlamydiales that are homologous to some genes found in several microbial eukaryotes for anaerobic metabolism. So these chlamydia seem to be capable of anaerobic metabolism, something that was not really seen before. And here in this figure, you have pathways related to anaerobic metabolism and microbial eukaryotes, and the colors represent the identity of their ancestors, like what kind of organism they originate from in eukaryotes. So purple proteins here, the purple circles, and so yes, this proteins seem to originate from alpha proteobacteria, so indicating that probably they come from the original mitochondrial ancestor. And then additionally, there's several genes that were found in this Anoxychlamydiales, potentially pointing to a scenario where a chlamydial ancestor exported these genes, transferred them either to a eukaryote, a bona fide eukaryote, and then transferred it to additional microbial eukaryotes, or potentially to the lineage present before the common ancestor of all eukaryotes, and then was inherited vertically, perhaps with some additional horizontal transfers. So this really comes to show that there's a lot of diversity that we don't know yet about, and they hold very interesting potential information for evolutionary scenarios. And in this case, this even raises the possibility that the origin of eukaryotes was more complex than previously thought, perhaps involving even more lineages than the two that we already know about. There's more work that we are doing to try to uncover potential origins from chlamydial genes into eukaryotic into the eukaryotic domain, and hopefully there will be more information about this coming out soon. But indeed, yeah, there's a lot that we don't know about the tree of life, and it is mostly sequencing studies and follow-up experimental studies that are showing that the diversity that exists in our planet is not only tremendous and holds a lot of information for processes that we thought we more or less understood, and it really happens to be that there's a lot that we can still say about them. Right, so this was everything I wanted to present, and I really would like to thank everybody for your attention. Thanks.